We often think that our risk of lots of diseases in life is determined by a combination of environmental factors, external things like lifestyle choices, and genetic factors, the genes we inherit from our parents. Whilst this is generally an acceptable way to understand our risk of developing some diseases, sometimes it's a bit more complicated. In particular, when we think about the role of epigenetics. Epigenetics looks at how external factors from our environment can actually change our gene activity without even changing the DNA base sequence of our genes, which are the basis of genetic inheritance. These changes in how genes are expressed can contribute to an increased risk of developing certain diseases, and they can also be passed down generations. Even the environment we are exposed to in the womb can alter the activity of our genes via epigenetics. These alterations can have long-lasting effects on our adulthood health and risk of disease. One of the key aspects of our womb environment that can influence this is maternal nutrition, i.e. the quality of our mother's diet. Our understanding of this phenomenon grew when studying the Dutch hunger winter that happened at the end of World War II. During this famine, rations were as little as 400 calories a day. When looking at the adults born around the time of this famine, scientists noticed that they had higher rates of obesity and type 2 diabetes. Along with other studies, this has suggested that poor maternal diet during pregnancy increases the risk of the baby developing metabolic diseases in adulthood. So how do we actually explain this trend? Well, lots of research has pointed to epigenetics as a possible explanation. The stress due to the baby not getting enough nutrients causes epigenetic changes to certain genes. These modifications don't change the DNA base sequence, but involve processes like adding methyl groups to the DNA or modifying histones, which are the proteins that DNA wraps around. This can cause changes in the expression of the genes and ultimately increases or decreases the production of the proteins the genes encode. These changes in protein activity can alter metabolic activity in the baby before it is even born. This allows the baby to adapt to the lower nutrient levels and increase its chances of survival. This sounds like a really helpful process, and it is when the fetus is developing. However, these epigenetic markers are permanent, meaning that as the baby grows to be an adult, the metabolic activity remains modified. If the adult no longer experiences nutritional stress, the altered gene activity can do more harm than good, by increasing the risk of certain metabolic diseases like type 2 diabetes and obesity. Truthfully, we still don't know very much about this concept. We need further research to tell us more about which specific genes are altered and by which mechanisms. As we research this more, it will contribute to our understanding of very prevalent and serious diseases like obesity and type 2 diabetes. Also, it will help us develop more new drugs and therapies that can help us treat these diseases. So, understanding how epigenetics influences our risk of developing certain diseases is a really important research area. My name is Amy Pagnam and I study Biomedical Sciences at the University of Manchester. My degree allows me to learn more about how our bodies work, what happens when it goes wrong, and how we can treat this.